right, let's get started. Hi, everyone. Today, we're going to have as our guest Joshua White, who I met a few months ago when I had the pleasure of being in Rotterdam. Joshua is a postdoc at the laboratory of Martijn Schonewille at Erasmus Medical School in Rotterdam. And he was a student that got his bachelor's degree from the University of Texas in Dallas in neuroscience, then moved to New York to the Albert Einstein Medical School to work, to do his graduate work with Roy Sillitoe, and then, as I mentioned, went to Rotterdam to do a postdoc. Today, he's going to talk to us about cerebellar modules. Thank you, Josh.
Yeah, thanks for having me. Thanks, everyone, for joining. I just want to show some recent data we've accumulated regarding, indeed, cerebellar modules and their relationship with movement control. So I'm going to jump in by starting with the reductionist framing that I keep in the back of my head, which is that I think that neurons have two primary features. One is how they are intrinsically, which is obviously a very loaded umbrella situation, and then who they talk to. And I find it quite interesting to think about how complex it is to get this right, to really make sure that the right neurons are talking to the right neurons. And I think that's a really hard problem for the brain to solve. And I think an interesting place to investigate that problem is in the cerebellum. So here is the sort of cerebellum that I study. This is the mouse cerebellum just back here. It's got all these folds called lobules. And if you were to cut a chunk of it off, it would look something like this. And you would see that the cerebellar cortex is really just a nice folded sheet of Purkinje cells. There's this nice single layer of Purkinje cells that go across the whole of the cerebellum. And around the Purkinje cell is built this complex circuitry, a relatively repeated circuitry, although there are differences from region to region. Purkinje cell receives input from the climbing fiber, a direct complex input, and then it receives an indirect input from mossy fibers via parallel fibers from the granule cell, and they project down to the cerebellar nuclei. The vestibular nuclei as the sole output of the cerebellum. And you can kind of see from this schematic, but I want to really highlight the Purkinje cell has this interesting feature, which is that we're kind of used to this beautiful, elaborate dendritic tree, but it's a planar neuron. So if you turn it 90 degrees, it will essentially just look like a line. And you think maybe that's a little sad. You kind of lose some of the beauty there. But it actually allows for another type of beauty of the cerebellum, which is if you stain for the right type of proteins, you can see this very fascinating striped organization of the cerebellar cortex. So this is now quite an old picture from even before I joined the Sillitoe lab of the expression of aldolase C and PLCA beta 4. And between the two of these antibodies, we can hit every Purkinje cell in the cerebellum, but no Purkinje cell will express both of them. And this organization is not just useful for making pretty pictures, it's actually a fairly fundamental element of how the cerebellum is structured. So if we look closely at the cerebellum, you'll see that so many things will mirror this striped organization that we see with aldolase C. And aldolase C was discovered many years ago. It's also called zebrin. It was originally by Dr. Richard Hawkes called zebrin. And so we still will kind of colloquially use that term zebrin. And you'll see that sometimes it's actually indicated as zebrin negative or Z negative, or zebrin positive, Z positive. And a while back, before I joined the Schonewille lab, they were recording from Purkinje cells in vivo. This is a flattened map of the cerebellar cortex with the aldolase C or zebrin map placed over it. They would record from Purkinje cells in vivo and then map the location of those Purkinje cells onto this zebrin map. And notice that if the cell was zebrin negative, it tended to fire at a higher rate than if it was zebrin positive. And a lot of theory actually falls out of that relatively simple this little graph here. A lot of theory falls out of that. It's quite fascinating. You can imagine that Purkinje cells that sit at a higher level, it's much more easy for them to decrease their firing. Purkinje cells that rest at a lower firing rate, it's much easier for them to increase their firing relatively. And that's indeed what the theory currently holds, that in cases of learning, that the cells that sit up here are more likely to pause their firing, and the cells that sit down here are more likely to increase their firing. And we'll get more into the evidence for that as I go. But in addition to these intrinsic properties, these molecular differences, these physiological differences, these striped this striped organization of Purkinje cells is also linked with differential input and output connectivity. So for instance, this is a study from a number of years ago from a colleague here at Erasmus, Tom Rauchak, and do not assume that I'm pronouncing that correctly. He has a very difficult name. But he injected rabies tracer into single muscles of the leg. And off the top of my head, I can't remember if this one comes from tibialis or the gastrocnemius, but regardless, you can imagine the number of synapses away that muscle is from this Purkinje cell, and you still ultimately end up with this beautiful band of Purkinje cells within the cerebellum, which means that potentially you have this discrete organization that is carried on and maintained many synapses down the way, all the way to the muscle. And the same goes for inputs to the cerebellar cortex. The climbing fiber is a well-known one. Climbing fibers will target these bands of Purkinje cells, but even the mossy fiber will, if you trace from the spinal cord, will land in the vermis in this nice banded way. So there's innumerable ways that you can find this pattern within the cerebellum, and I've been interested in it for a number of years now. So I want to just show you some evidence from the literature of the different ways in which this organization might kind of show up behaviorally. One way is that they tend to be organized into which behaviors they're involved in and how they respond to the learning in that behavior. And so the first example I'll show you is in eye blink conditioning. Eye blink conditioning is when you take a neutral cue like a tone or a light, and you pair that with the air puff to the eye, which will induce an eye blink. And over time, the animal will learn to associate that neutral cue with the air puff, and they will learn to blink to now the neutral cue. And so what you see here is after training, this is the eye blinking. As the eye goes, as the line here goes up, that's the eye blinking. And you can see that the eye begins to blink before the air puff occurs, which is at this line here. And if you record from Purkinje cells that are associated with this behavior, you see that they dip their firing here. And so that's what I was talking about with the Purkinje cells that tend to pause their firing. And you can see that the peak, or maybe the trough of that dip, it correlates with the onset of that conditioned response of now the blink to the neutral cue. So the areas that tend to be associated with eye blink conditioning tend to be zebra negative, outalase seed negative areas. Those are the cells that sit at that higher level, and they also tend to pause their firing in the case of a learning event. So if we look now at a different form of learning that's cerebellar dependent, the vestibular ocular reflex. This is a reflex that will turn the eye in order to maintain a stable visual field. When the vestibular ganglion is activated, or when there's some sort of kind of vestibular event, it allows the visual field in front of you to remain stable, largely controlled by an area called the flocculus, which is largely zebrin positive. And if learning is taking place in this system, it tends to be a learning in the upward direction, where simple spiking tends to increase when that learning is occurring. So here you see two different regions of the cerebellum, two different types of Purkinje cell based on the outalase C expression, and two different ways in which the learning is expressed electrophysiologically. So another interesting finding you can find in the literature is when you activate Purkinje cells, you can drive movement in different ways. So this is actually from the Medina lab. They used activation of the inhibitory basket cell in order to reduce firing of the Purkinje cell. So here's optogenetic activation of a basket cell. You can see it increase its firing. Here's what happens to the associated Purkinje cell. It pauses its firing. And if you do that with many Purkinje cells, you can induce a movement, in this case, an orofacial blink type movement. And that movement is time locked to the optogenetic stimulation. It's time locked to the pause of that Purkinje cell, and the duration will last as long as the optogenetic stimulation lasts. So you have the stimulation, which can drive the movement. A year after that, the Otis group found the same thing, this time by direct inhibition of Purkinje cells using archirhodopsin. And they find, again, a movement, this time a limb movement, time-locked with the inhibition of Purkinje cells. But they also use channelrhodopsin to excite Purkinje cells. And interestingly, they find that movement occurs time-locked to the end of that excitation. So during this little pause in the firing after the relative increase of Purkinje cells, that's when you see the onset of the movement. And this is all in contrast. These optogenetic stimuli were done in the vermis, paravermis areas that tend to be zebra negative. This is in contrast to what you see if you activate the flocculus. If you put channelrhodopsin and Purkinje cells at the flocculus and you activate them optogenetically, you see movement time-locked to the onset of the increase of Purkinje cell firing. So in this case, an increase in firing induces the movement. In the case of the more zebra negative areas, a decrease in firing induces the movement. And last is one more feature, which is the differences in learning rules of these populations. This now goes back to 2005. Wadish and Jar used an LTD slice protocol to show that it was more readily, you were more readily able to depress Purkinje cells within lobule 3, tends to be a zebra negative area, then in lobule 10, tends to be a zebra positive. We showed that again recently using an EAT4 mouse all around the cerebellum, but now specifically just looking at whether or not the cell was zebra positive or negative. And another paper that's of note came from the Raymond lab, where they used the actual timing of the relationship between stimulation of climbing fibers and parallel fibers, and found that based on the relationship of this timing, you can induce different plasticity events in different parts of the cerebellum. And in this case, for instance, with simultaneous parallel climbing fiber activation, you can cause LTP in the flocculus and LTD in the vermis, showing that the exact same sort of stimulus will induce different plasticity events in these populations. So all of this kind of together leads me to wondering what are the kind of behavioral benefits of having a system like this, having this kind of organization, having these sorts of the same sort of neuron with these very different intrinsic properties and different learning rules. And that leads me to wanting to know what are the overall behavioral function of these modules. So these Purkinje cell populations are part of these cerebellar modules. You have the Purkinje cells up in the cortex. They project down to cerebellar nuclei or vestibular nuclei, and they receive input from a specific part of the inferior olive to make this lovely little parallel organization of the cerebellum. So I think that in order for the kind of the modular control of movement that I have in my mind to be true, there are many necessary pieces, and I'm going to show you evidence for a few of them today. So one is that they are part of these discrete pathways, that the information is carried forward in a parallel discrete manner. Another is that the modular activity is correlated with specific features of movement. So I'll try to show you an example of that. And lastly, I'll show you a bit of a more recent project I've been undergoing, showing that there's differentially active during a complex movement. So I'm going to start by going to the flocculus. This is a brain structure important for compensatory eye movements I mentioned before, such as the vestibular ocular reflex, which allows for when this man rudely jerks this woman's head around, she can maintain her eyes looking straight ahead. So that's based on the vestibular ocular reflex. You have the ability to move your eyes in the opposite direction of the vestibular movement in order to not lose sight of lose the stability of the visual field. So if you were to do something crazy like bike down some stairs, you could at the very least see a stable view of the world as you do it. And the flocculus is important for learning this reflex and for maintaining this reflex. We can actually monitor changes in the gain of this reflex by monitoring neurons of the flocculus. And so we've been studying the flocculus for quite a long time within the Schonewille lab. And there's been work recently showing that the purely zebra positive nature of the flocculus may or may not be so. It's a little complicated, but at the very least, we've known for a long time that it's difficult to really see different regions of the flocculus. If you look at a coronal view of the flocculus, you look at the posterior end stained with zebrin 2, you look at the anterior end stained with zebrin 2, and you don't really notice much difference. If you look at a space between, which happens to catch a boundary, you can see that there's actually what looks like a dark zebrin area and then a lighter zebrin area in the more anterior side of the flocculus than in the posterior. And there's actually something to this. And if you look at different markers, you can find a more binary example of these two subpopulations of the flocculus. And here we found a marker called KCTD12, which is strongly expressed in the posterior flocculus and then not expressed in the anterior flocculus. And so we thought that this might be kind of neat to focus in on this relatively small structure with these apparently two different subpopulations. If we now look at a sagittal view, you can see its relationship directly with autolase C, where you have this KCTD12 negative region in the anterior and positive in the posterior, and you have a very dark autolase C or zebrin positive in the posterior and a lighter autolase C in the anterior, and that's schematized here. So we thought this could be interesting, but before we kind of embarked upon investigating these subpopulations, we wanted to really make sure that they were different. And we knew from previous work that you could find differences using acute cerebellar slices. So that's what we did next. We would slice the flocculus, record from Purkinje cells within the flocculus and block all synaptic input and just record intrinsic activity. Label the cells and then identify them as either KCTD12 positive or KCTD12 negative. When we look at just the intrinsic cell attached firing rate, you can see that the KCTD12 negative tend to fire at a higher rate than the positive, but not as high of a rate as, for instance, the classically zebrin negative lobule 3. And so there's actually kind of an intermediate level that exists within the flocculus that correlates with that kind of intermediate level of autolase C. So we're going to complicate the stripe marker expression just a little bit, and maybe cast doubt on the purely zebrin positive and negative, but only a little bit, only in some areas. If we patch the cell and record whole cell and induce action potentials by injecting current, we see the same thing. We see that the lobule 3 cells fire at this high level, and then the KCTD12 negative floccular Purkinje cells fire at this intermediate level, and the KCTD12 positive fire at the lowest level. And these are the slowest firing Purkinje cells that I've recorded from. Lobule 10 sits right here in the middle with the two surrounding it. So we thought now that we have this nice subpopulation that are intrinsically physiologically different from each other, and we wanted to start investigating that a little bit further. So to do so, we needed some additional tools, and we needed the ability to genetically target these subpopulations. So we started looking for Cre-lines and found a couple that are fairly useful. So here is just an image of the whole cerebellum going from posterior down to anterior. Here you could see KCTD12 has a striping pattern very similar to autolase C. And I've added in HSP25 for any big fans of stripes that want the additional piece of information, but also to show the comparison with CAMK2 Cre, which is the first Cre marker I'll highlight. CAMK2 Cre is this is a transgenic line that in somewhat of a unusual way just happens to be expressed in this pattern within the cerebellum. This is a function of the transgene. And the nice thing is that within the flocculus, it's expressed in the same area as KCTD12. And I'll show you a highlight of that in just a moment. The other Cre line that we use is KCNG4. KCNG4 is expressed in a KCTD12 negative manner, so these two are complementary to autolase C for the most part. You can see the relationship here. So in the anterior vermis, where autolase C is predominantly negative, KCNG4 is predominantly positive, for example. So just a big index of all of those. But now we will highlight the flocculus. So here is the posterior coronal flocculus, anterior coronal flocculus, the expression of KCTD12 in the posterior, the expression of CAMK2 Cre in the posterior, and the expression of KCNG4 Cre in the anterior. And here's a basic schematic showing its relationship between the three and also with autolase C. So with autolase C, you have the classically negative autolase C that would exist in, for instance, the anterior vermis is going to be strongly KCNG4, and KCNG4 will also be expressed in the moderate autolase C, but never in the KCTD12 region here. Okay, so we use these Cre-lines for a couple of reasons. The first is to do some viral tracing. So we inject into the flocculus in a Cre-dependent way and trace from these different regions. So this is an injection into the CAMK2 Cre, the posterior flocculus, and we're just looking for the ultimate termination of Purkinje cell fibers within the vestibular nuclei. And here what you can see is a lot of termination within the prepositus hypoglossy, the medial vestibular, but not very much in the spinal, lateral areas of the vestibular nucleus. On the other hand, if we inject into the flocculus of the KCNG4 Cre, which is in the anterior flocculus, we see very little tracing into the medial vestibular and the prepositus, and predominantly tracing into the lateral vestibular here. So we actually can see these nice discrete pathways, which we can summarize here with the two halves of the flocculus shown here, the KCTD12 positive in the back and the KCTD12 positive in the back here and KCTD12 negative in the front. And we know from previous literature that there's actually 4 zones across the mouse flocculus related to either vertical or horizontal movements, or response to vertical or horizontal visual movements. And so what we think is that they're actually split to where each of those zones has both a KCTD12 positive area and a KCTD12 negative area, suggesting that different subpopulations are contributing to the same ultimate movement. But they're projecting in this discrete way down to these different targets within the vestibular nuclei. So this is our interrograde tracing via these Cre-lines. We also wanted to know if there was any breakdown of which types of neurons were targeted within the vestibular nuclei, and for that we used rabies tracing. The basic scheme for that is to inject a helper AAV virus, which will allow for the infection of the rabies and also for the transsynaptic tracing back one synapse for the rabies. So we inject the virus, the AAV, wait 2 to 3 weeks, and then inject the rabies also into the vestibular nuclei. And then after about a week, that will trace back to Purkinje cells within the flocculus. So that in general will look like this. This injection happened to target the medial vestibular, and it traced back to this Purkinje cell here in this KCTD12 positive area of the flocculus. Okay, so we used this basic scheme, and we wanted to see maybe one region or the other projected primarily to inhibitory neurons within the vestibular nuclei. So the first injections we did were in GAD67 Cre or GlyT2 Cre lines, which will target GABAergic, glycinergic, inhibitory neurons within the vestibular nuclei, and we could see no systematic breakdown. We saw Purkinje cells traced back into all regions of the flocculus in every injection that we did. So here you can see the spread of the different injections that we performed. Here are 4 different injections, and then you can see where those injections traced back to within the flocculus. So we didn't see a clear relationship between floccular subpopulation and GABAergic or glycinergic targeting. So next we injected into excitatory Cre, the VGLUT2 Cre, and here's the spread of the injection from different injections in the vestibular nuclei. And again, we couldn't see any systematic difference within the flocculus. We saw tracing back to both sides, and so we thought, well, that's we didn't really know what exactly to do with that, but that just means that both areas target both inhibitory and excitatory neurons within the vestibular nuclei. So next we looked for any possible subpopulation that we could find within the vestibular nuclei, and we found that the SST Cre line would allow us to target a subpopulation of neurons within the medial vestibular and prepositus area of the vestibular nuclei. And in that case, if we inject and perform our rabies tracing, here we see a isolated tracing back to only the KCTD12 population. And so that's what you're seeing up here, a KCTD12 positive Purkinje cell that was traced back from an SST tracing. So here we can see that while there's not a clear organization of an inhibitory or excitatory, there is the existence of discrete pathways that are coming out of this subpopulation difference within the flocculus, and presumably many, many more. SST Cre is not going to be special in this case. There's going to be subtypes within the vestibular nuclei that are receiving these subpopulation inputs from the Purkinje cell. So we were happy to see that, and we now proceeded to my next question, which is the interaction of modular activity with specific movement features. So to investigate that, we return to our Cre-lines, we return to our CAMK2 Cre and our KCNG4 Cre, and this time we use those Cres to activate the expression of chanorodopsin so that we could stimulate Purkinje cells, activate Purkinje cells. And just as a reminder, in the flocculus, activation of Purkinje cells drives movement. And so the scheme looks something like this. We put the animal into a head fixed setup. We aim a fiber optic at the flocculus in mice that express chanorodopsin either in all Purkinje cells or in just Purkinje cells within one of those subpopulations. When you turn on the light, you induce a movement in a nasotemporal and an upward-downward direction. The horizontal movement will always be larger than the vertical movement, at least in our hands. And it'll have this kind of general shape. You'll have this rapid onset and then this slow movement afterwards. So we did this experiment in all three of our lines, either in the KCNG4 Cre or a PCP2 Cre to target all Purkinje cells, or the CAMK2 Cre, and we found that we could really reliably evoke eye movements in the KCNG4 and the PCP2, very little in the CAMK2 Cre. But then the eye movements evoked between the KCNG4 and the PCP2 Cre were always reliably different between all mice, where you tended to see that the onset of the movement was the same between the KCNG4 and the PCP2 Cre, but then the movement during the duration of the optogenetic stimulation was different. So what I'm showing you here is 3 different durations of optogenetic stimulation, 200, 500, and 1000 milliseconds. And then here, the eye movement traces in both the horizontal and vertical directions of all 3 mice. To really see the difference, we broke down these movements into 4 components. There's the drive, the onset of the movement, the hold portion, which is during the duration of the optogenetics, the release of the optogenetic stimulation, and then the recovery phase back to baseline, back to where the eye was before movement. So if we look at those 4 components, the drive, hold, release, and recovery, we can see that there's no difference between the KCNG4 and the PCP2 in the drive or release. And I would term those as the fast components of this behavior. But we do reliably see at the longer durations differences between KCNG4 and PCP2 in the hold and in the recovery phase. So the slow components of the movement are the ones where the differences really come out. And the difference of the CAMK2 is just not ever very clear that the movement was really induced with activation of CAMK2. So we believe that subpopulation is not very involved in the actual induction of a movement or the potential onset of a movement, whereas the KCNG4 very much is, but the KCNG4 is predominantly involved in the rapid change of the movement, and less so in these kind of more the slower phases of a movement like what we see in the PCP2 induction. So this is the general feature of what we see with the modulation of the activity. And I'm going to move on quickly to show you my last piece of these 3 here, which is that modules are differentially active during a complex movement. So I've been interested for a while in the fact that the evidence for differences of Purkinje cell subpopulations related to behavior are coming from very different types of behavior, eye blink conditioning versus vestibular ocular reflexes. These are very different types of behavior. They're doing very different things. And I wanted to see if we could drive a behavior that would activate both subpopulations during the same behavior, during the same movement, and see if the upbound, downbound phenomenon holds up in that case as well. And so we've developed a new classical conditioning process. We're calling it brace conditioning, in which we show a light. The animal is head fixed above a wheel, and after 250 milliseconds of showing the light, we just move the wheel very briefly beneath the animal, just enough to cause them to make a reflexive bracing movement. And so this was actually very early EMG recordings that I took when I was first testing that this behavior would work. And I placed the EMG wires subcutaneously down the trunk of the animal. And in the beginning, what you can see is no movement in response to the light, only movement in response to the wheel moving. But after some training, the animal begins now to make a movement, predictive movement before the wheel, now to the light. So this is my learned movement, or the conditioned response, if you will, to the conditioned stimulus that is the light. So I liked the idea of now having a timed whole body movement, which should activate large regions of the cerebellum because it's activating so many muscles. And we should be able to now investigate the role of different Purkinje cell subpopulations in the performance of a singular behavior. So here's an example of what this looks like. This is a CS-US training trial. So what you'll see here is this is a copy of the light that the animal is looking at, just for our sake for this video. So you'll see the light turn on, and then you'll see the wheel move below the animal. And this is slowed down to make it easier to see. So this is a training trial. It's a very small movement to the wheel, but it's enough to induce a reflexive movement in the animal. And then after training, I'm going to show you an actual time CS only. So we're only going to show the light and not move the wheel, and you can see what the learned behavior looks like. And so the animal now just makes this very relatively calm, actually, bracing movement in response to the wheel. If we look at the EMG activity of just a control mouse, you can see that on day 1 here in yellow, there's very little EMG activity preceding the wheel and a lot after the wheel. Day 2, you see the beginning of a hint in this animal of some movement before the wheel. And by day 3 and 4, the animal is now readily making movement before the wheel. These are in paired trials. And then this is just a raster plot of the movement onset according to when the EMG waveform crosses a threshold. And I just plot that time point here, and you can see that move forward. In CS-only trials, you can see that the animal really doesn't respond much on day 1 and 2, but now it starts making a reliable movement prior to when the wheel would take place. This is when the US would occur if it was a paired trial. And you can see that the animal now moves the movement onset up in time, and it becomes nicely time locked to about, it's usually about 110 milliseconds or 120 milliseconds after the conditioned stimulus. I also checked this in a mutant animal. This is a mutant animal that we've used for a while within the department here. We delete a protein called PP2B exclusively from Purkinje cells, and it reliably induces learning deficits in different studies. And so we wanted to check to see if there was a cerebellar component to this behavior. And this is a control condition where you see the animals readily learn to make the proper condition response. And the mutants really are quite scattered in their response. If they do show what could be a conditioned response, which I define as any movement within the 500 milliseconds after the CS, it's really quite random in time and not very reliable. Here's the quantification for the group of mice. You can see the control mice nicely move forward in time over training days in both the paired trials and the CS-only trials, whereas they're significantly better than the mutant mice in both of these cases. The conditioned response percentage increases over the days more so than in mutant mice, and the standard deviation of this movement onset gets smaller with training as the timing becomes more precise, and that does not occur in the mutant animals. There is still something of a potential response there, which I find quite interesting, but obviously significantly worse than control mice, suggesting that indeed the cerebellum is contributing to this behavior. So I now have this nice movement that I can reliably evoke by just turning on a light. And so now I just go around and record from Purkinje cells while I do this, keeping in mind this previous finding that Purkinje cells of different subpopulations show these different intrinsic properties. I wanted to see if they would also show a difference in their response to this behavior. So the way I identified them, I used one of two ways. I recorded the cell and injected neurobiotin in some cases. So here's an injection within an EAT4 mouse, which is expressed at the same pattern as Zebrin2. Here's an injection in a KCNG4 Cre mouse, which is obviously the opposite of Zebrin2. And here's just the mark to see where my Purkinje cell was. But actually, I took advantage of an additional quirk of the KCNG4 Cre, which is that in addition to being expressed in Zebrin-negative Purkinje cells, it's also expressed in basket cells. So it's expressed here in basket cells, which means that if you shine a light over this set of cells, you're going to activate these Purkinje cells and you're going to inhibit these Purkinje cells, which is actually the most ideal situation for optotagging subpopulations of Purkinje cells. And so that's exactly what I do in vivo. I put a fiber optic into my glass recording electrode. I start recording my Purkinje cell and I give a 50 millisecond pulse of optogenetic stimulus. And I can see, is this a KCNG4 negative cell by showing me that it's inhibited, or is this a KCNG4 positive cell by being excited? And I can in real time classify what type of cell I'm recording from. So just as an example, I will show you a cell that I was recording here. I turn on the optogenetic stimulation. I see a reduction in the firing, which means I can classify this as a KCNG4 negative cell. In parentheses, autolacy positive. It's, I know, probably hard to keep all these things in your head, but this is now a Zebrin positive Purkinje cell. So I've identified it with optotagging. If I now present my behavioral stimulus by showing the light, the conditioned stimulus, I evoke the condition response shown here with the EMG activation. This Purkinje cell increases its firing in response to the conditioned stimulus. On the other hand, if I'm recording a Purkinje cell and I flash some optogenetic stimulation and the cell increases it, then I can classify it as a KCNG4 positive cell, autolacy negative. And in this case, if I show the conditioned stimulus, I see a pause in that Purkinje cell's firing. So if I kind of group many Purkinje cells together, and this is a good deal of the data set, this is the firing rate when I give the optogenetic stimulation. It's a 50 millisecond pulse, and you can see that they either are classified as KCNG4 positive by increasing their firing or KCNG4 negative by decreasing. That same population, if I show the behavioral stimulus, will now show the opposite results. The ones that were inhibited will now be excited, and the ones that were excited will now be inhibited by the behavioral stimulus. And this is the average of the EMG response for those two at the time that I recorded those two populations. So you can see the time locking of the simple spiking to the movement. So in both cases, either response is time locked to the activation of the EMG. And one final note before I stop, an interesting point that I find is that I see the existence of a complex spike in both cases. So whether or not the cell is going to increase its simple spiking in response to the movement or decrease its simple spiking, often you'll see, and not on every trial, but on many trials, you'll see a complex spike that just precedes the movement. And here what I've done is plotted all of my first complex spikes after my behavior versus my onset according to the EMG activity. And you can see there's no real difference between these two subpopulations of Purkinje cells. And on the whole, the complex spike occurs just prior to the movement, since we're underneath the unity line here. So that suggests that regardless of the cell type that we receive a complex spike, But because of the intrinsic differences of these subpopulations, we see a different simple spike response. So I'll stop there and conclude that Purkinje cell populations are indeed part of discrete pathways, that differential activation of Purkinje cell subpopulations can drive different components of a movement. Mice can readily learn a whole body movement using brace conditioning. The simple spike response correlates with the molecular identity, and regardless, there appears to be a complex spike that can occur just prior to the onset of the movement in response to the learning. So I'd like to acknowledge quite a number of people, obviously the Schonewille lab, and especially students who have helped me with these different projects, and then the neuroscience department in general, and some of the funding sources that I have. And I'd be happy to take any questions.
Thank you so much, Josh.
Of course.
Yeah, let me start with just a comment about that beautiful task that you presented. You know, I was thinking that the response to learning, I suppose, if the direction of wheel rotation is consistent with respect to light onset, then you might expect a difference between if the animal has learned a rotation forward versus a rotation backward in terms of the motor response. So not just the bracing, but a direction-specific learned pattern. I don't know. I wonder if I'm sure you've thought about that. You've looked at your data from that perspective.
Yeah, I mean, it's interesting. In the beginning, the mice make fairly kind of random movements in response to the wheel moving. I would say it takes them a little while to really get like this kind of response. This is after days of training, and their movement becomes really reliable and kind of subtle. But in the beginning, it's quite wild. But it would be interesting if now I taught them to move in the other direction. It would be interesting to see exactly what that movement would really even look like. What is the ideal bracing for a movement in the other direction? But yeah, indeed, I'm not sure if there would be a difference there. The question is if we would really be activating muscles in a different way. Because what I think is happening is that we're activating muscles that have different needs for its neural input, in which case you use both subpopulations of Purkinje cells, and then you need both of their directional responses in order to activate the type of muscle correctly. So I don't know if the direction would change it if the ultimate movement were similar, I suppose, and if it activated a similar set of muscles, if that makes sense.
The behavioral literature, these are called postural responses, and what typically people do is that they have them stand on a platform, and the platform shifts forward and backwards, and the response is, of course, very specific to the input that the subject receives.
Indeed.
Jennifer, I see. Raise your hand. Go ahead, Jen.
Jennifer Raymond, can you hear us? I don't know.
Oh, hi, sorry. Several of us are watching together, and I didn't want to get a feedback thing by looking up my
problem.
PC. Anyhow, thanks for that. Awesome, interesting work. I'm very interested, especially in the different time courses you get with the two subpopulations in the flocculus. I love that you're dissecting that, but I'm not understanding exactly why you're ignoring that the neurons in the flocculus can drive eye movements with decreased firing, right? So like if you record, they increase their firing for ipsiversive, we call it, so nasotemporal, and they decrease their firing below baseline for the opposite direction. And if we activate with CHR2, we get an eye movement in one direction. If we inhibit with ARCH, we get a movement, we initiate movement, but in the opposite direction. So you kind of emphasize the upward going part of their response, but they have the opposite. And you mentioned the Parnas paper in my lab. We do get LTD if we account for the climbing fiber delay. So what do you think about, is there What do you think about the oppositely directed responses in the flocculus?
Yeah, I think they are. Yeah, the modulation of the Purkinje cell firing can lead to responses in both directions. We've always been much more successful in inducing eye movement with excitation, and the relative eye movements with inhibition are really quite small in comparison. And I don't know if that's a feature of the muscle, the need for the muscle to move in the other direction, but I don't know if you see that there's a difference in the amplitude of movement that you can get with excitation or inhibition, but that's what we've been seeing.
Yeah, there could be, it's hard because we have had CHR2 or ARCH. So, but there may be a little asymmetry even in the recordings physiology. But clearly the whole circuit for eye movements is set up with this push-pull control, and that you can get an eye movement at almost any point by increasing or decreasing firing of the neurons. So the whole system is really set, it seems, to go with a little increase or decrease on one side that upsets the balance between the kind of push-pull control from the two sides of the brain. Yeah,
it's interesting. In our hands, we just haven't seen as readily a movement with inhibition as we have with excitation. But I agree. I mean, it makes logical sense, and we do see a little bit of movement, but it's always been much more significant using excitation rather than inhibition. So. Yeah, I think Amir, you had a question as well?
Yeah, thank you for your presentation. That was fantastic. And sometimes I feel I can inspire in my work. In one of your data, I'm interested to know how you had some problem to analyze your result. You have, so probably in some places you had very narrow, very narrow differences between your data. And signifying them and finding the relation sometimes quite being the WEX part of the defining the finding the tuning the result. So in one of pages, I'm not remember 25 or 26. Can you change to
would it be the eye movement or the
it
body
was a
movement?
swarm plots. Can you remember a swarm plot in your data? Or
this one here?
Yeah, exactly. That's in the right bottom.
Yes. Yeah.
What those dots are representative of what they are showing us exactly.
Yeah. So this is the onset of the movement according to the EMG waveform crossing a threshold. So this would be essentially more or less the onset of when EMG actually, when a movement is actually beginning in this case. And so you see that over the learning days, the movement will creep forward in time, and now the movement will occur in response to the light and less in response to when the wheel would be. And this is just an example of a single mouse, but the timing gets more locked into this particular time here if you look at many mice.
So what was your agony in here? What was challenging for you to draw such kind of understanding for yourself? How did you resolve this? I just want to get inspired. How you get over that problem that I can make understand people and convince this is true?
So I've done it in a couple ways. So I'm only showing you the results from the EMG in this talk, but we've also recorded video of these animals and then analyzed the movement with deep lab cut to actually check for movement in that case. And I am a little I want it to be quite convincing, so that's why I wanted to kind of duplicate those findings, and I will show that when we submit this paper. So the onset of the EMG, I think, is very nice in terms of the temporal resolution and being able to actually see at a very, very high sampling rate the EMG activity, but EMG is by itself very messy. But the deep lab cut is obviously we're hindered by the sampling rate of the video, but it's a little bit it also has a cleaner element to it because you're just watching the individual frames. And then in order to actually identify when this is, I just went trial by trial and checked the baseline of each trial and then waited to see when the EMG crossed a standard deviation above that baseline. But getting that exactly right and filtering the EMG right was quite a challenge in the beginning, but at this point it's I think pretty straightforward.
Just this kind of things are really challenging. So I'm pretty sure I have sometimes in my data I have some differences, but quite extracting and putting them on a graph to show that differences sometimes quite. Thank you so much.
Yeah, of course.
Yoshiko, you had a question.
Yeah, thank you very much for the great talk. So I have a question about complex spike. Zebrin positive and zebrin negative, is there any difference in the complex spike, like spike rate duration, like pause duration of the simple spike, or the baseline firing rate?
Yeah, there is a difference there. Let me see, how do I just look at the whole slide view here? If I go back to this one, so this is only one piece of the figure from this paper, and other people have actually looked at even more detail at this. But in this case, you can see that there's even a difference in the pause after the complex spike of the pause and the simple spiking. You can see that sort of difference. There's probably differences in the size of the complex spike, but that's quite difficult to really be confident of unless you're maybe doing whole cell patch clamp of the Purkinje cell. There's at least a difference in the effect of the complex spike according to the Zebrin identity.
So,
and you asked about the rate. The rate is also matches the same thing as the Zebrin negative and positive, where there's tends to be higher in the Zebrin negative and lower in the Zebrin positive, actually. Yeah, and then these I'll show you one example. These differences are quite come about quite early. So this is from a paper that we published a couple years ago, and we looked for spontaneous firing in vitro and just recorded cell attached, and we see that the difference emerges at about a week and a half. And this same paper, we also examined in vivo recordings from very young animals as well, and we see the same difference in complex spiking from a very young age as well. So I recommend, if you want to see further, you can have a look at this paper as well.
yeah, great. Thank you very much.
Yeah.
Thank you so much, Joshua. Are there any other questions?
I actually have a question.
Oh,
Hi,
hi.
Josh. That's a great talk. Thanks so much.
Thank you.
Hey, I was curious about your sort of take on the transsynaptic tracing experiments from the inhibitory and excitatory neurons of the vestibular nucleus. It seemed like the Purkinje neurons that are projecting to these two populations may be intermingled and share Zebrin identity. Is that an accurate interpretation there?
Yeah, I think so I mean, basically we don't see that one of these populations preferentially targets Zebrin positive or negative.
Right. So regarding that,
so excitatory or inhibitory, sorry.
Right. So regarding that observation, I was curious about sort of your take on that in the context of the hypothesis that there are these upbound and downbound modules that would sort of link into the inhibitory outputs versus the excitatory outputs. Could you just comment a little bit on that? Thanks.
Yeah, definitely. So I think that might get to the fact that we have each of these subpopulations is linked to these different behaviors, the horizontal vertical axis, for example, and the fact that they are often in the flocculus, as Jennifer was pointing out, there's kind of a scaling to some extent of the Purkinje cell response and the movement, and so to some extent makes sense that you would want both of them to have both an excitatory and inhibitory potential push and pull. But I think we would find, if we looked more specifically, that they would probably target excitatory and inhibitory if we weren't using the general GAD2 and VGLUT2, for instance. The SST in this case is a specific excitatory neuron, and that's, I think, where we're going to see the differences. And then the way that those neurons ultimately project down, I think that will be how you get the upbound downbound difference, depending on the behavior that you're actually influencing, if that makes sense.
Yeah, so then the prediction there is that you maintain the prediction that the sort of upbound Purkinje neurons are going to selectively converge on specific subpopulations of neurons, and the downbound a different population, and not mix.
No, so I think, yeah, exactly. I don't think they will mix. I think they maintain their discrete tract ultimately, but I think they can influence the downstream target in either case, and by both targeting inhibitory or an excitatory, as long as it maintains that specific pathway. That's what I would hypothesize.
Thank you.
Oh, Trace.
Hey, Josh. Thanks. Oh, no. Thanks for your talk. I was just wondering, can you remind me, and maybe I missed this, but what's the relationship between the KCTD12 and the KCNG4? Are those overlapping, or are those different?
No, they are the exact opposite. So actually, I showed you in the very beginning that classic picture of AUDL-AC and PLC beta 4, and those are exact opposites. The KCNG4 and the KCTD12 are both slightly different from AUDL-AC, but these two are also exact opposites, actually, and that occurs throughout the cortex. It's an interesting phenomenon. It's hard really to tell, but there's actually these little places where you have some intermingled KCNG4 AUDL-AC bands right here, if you all can see that, but you won't see KCTD12 intermingling with KCNG4. So there's actually kind of additional agonist-antagonist markers that we've been finding, if you will. But yeah, so these two are complementary to each other.
Cool, thank you.
Mm-hmm.
So much, Josh. Any other questions? Wonderful to see you. Wonderful to be with you today. I hope you have a wonderful Tuesday. Bye.
Bye, guys. Thanks so much.